Good morning, everyone. Can everyone hear my voice?
Yes.
Yes.
All right, that's good. All right, thank you. So, welcome you all to today's webinar. What you are missing matters. Deliver food safety with Oxford Nanopore. Thank you all for joining us today. I'm Mengki, and I'm the moderator for today's webinar. Before we begin, a quick note on housekeeping. This session will run for about 30 to 40 minutes, and we will have a Q&A session towards the end of the webinar. If you have any questions during the webinar, please feel free to submit them in the chat box, and we will address them after the presentations. To ensure the best experience for all participants, please keep your microphone muted throughout the session. In today's webinar, we are excited to have Mr. Chi Hong as our speaker. He is the Field Application Manager at NextGene Scientific with over a decade of experience in his field. He will be sharing valuable insights on Oxford Nanopore technologies, its application in food testing, how nanopore sequencing enhance food safety, and comprehensive end-to-end solutions for the food industry. Without further ado, let's welcome our speaker today. Chi Hong, the floor is yours now.
Okay, thank you, Minty. Thank you for your kind introduction. So let me share my slide. Okay, so Minty, can you see my slide?
Yes, we can see your slides.
Okay, unfortunately, there will be some delay. Hopefully, it won't affect overall experience. So thank you all for joining today. So my name is Chi Hong. So today I will be sharing on the deliver food safety with Oxford Nanopore technologies. Without further ado, so first off, I would like to begin with a short introduction on the the manufacturer of the sequencing technology, which is Oxford Nanopore Technologies. After that, then we will go to using this particular sequencing technology to perform some of the food safety testing and application for the detection of food pathogen as well as the food authentication. Okay. So Oxford Nanopore Technology was founded since 2005. It's actually a spin-out company from Oxford University. So the key technology of Oxford Nanopore Technology is actually the use of the protein pore for sequencing. In year 2014, they actually launched their first sequencing device, which is the one that's showing here. We call it the MinION. And then in 2015, we have the bigger brother of MinION that we call ProMinION. The only difference of these two is that ProMinION will be able to produce more sequencing data and cater for larger genome sequencing purpose. So moving forward, then Oxford Nanopore has become a global presence as a sequencing machine offering. Okay. So currently we have over offices in more than 125 countries. We are serving more than that. And then the nearest one will be in Singapore. The platform of Oxford Nanopore technology can be used for the sequencing of genomic, which means that DNA, and then we can use it for the detection of epigenetic, the methylation, as well as to look at the RNA transcriptome status, for example, gene expression, so on and so forth. Currently in development, we are using the same platform, which the key technologies is nanopore, for the sequencing of protein as well as metabolomics as well, but that will be in few years time, currently not available yet. Oxford Nanopore technology can be used for the sequencing of DNA or RNA, so therefore it's suitable to be used for human genetic sequencing, cancer study, even plant, genomic, transcriptome, animal, and microbia. So it doesn't matter whether it's big or small, nanopore can be used for the sequencing of the genomic DNA or transcriptome. Okay. So as in terms of the field that are relevant, that actually suitable to use the sequencing technology, nanopore can be actually used for the healthcare clinical setting, for example, the hospital for diagnostic, for the detection of pathogen and all that. For agriculture, nanopore sequencing can be used for the detection of genetic modified organism or the mutation to generate GMO as well. And then for the food sector, it can be used for the food safety monitoring, which will be the focus of today, as well as environment biodiversity study, the surveillance for biosecurity, so on and so forth. So this is just to show you that the key technology of nanopore technology, which is actually the protein, showing here the nanopore. This is an animation showing the DNA strand passing through, and we are detecting the base that passing through that enable the sequencing and getting the information of the nucleotide. Here we have the two flow cell that offering by nanopore. The smaller brother, which is actually the mean ion, we have a smaller output, and we have the bigger brother that produce more data in comparison. Okay, so that will be it for the introduction of Oxford Nanopore technology. Now we shall move on to the usage of nanopore sequencing technology for food safety and food quality purpose. So food testing program involve oversight of the food itself and the environment of where the food is being produced and manufactured. So for example, the food production factory. So to monitor the food, we can actually use nanopore technology to monitor the presence of foodborne pathogen and also microorganisms that causing the spoilage of food, some indicator microorganism present, and as well as the food authenticity application. Okay. So our end-to-end sequencing approach accommodates different industry standards and needs as a wider QC quality control or confirmation testing stage to prevent or address safety or quality issue. So these are some of the four key offering from nanopore technology to provide insights to control contamination and prevent food fraud. Okay. So the first one, nanopore technology can be used for the sequencing of the pure isolate of, let's say, bacteria or fungus. We can perform whole genome sequencing. So this is useful for some foodborne outbreak surveillance purpose. It can also look for to give us better understanding on the probiotic or the yeast strain engineering purpose. So for the amplicon sequencing, we can use also nanopore to perform amplicon sequencing. This method is actually very useful for rapid foodborne pathogen detection because in order to get pure isolate, we will need approximately some enrichment stage, some culturing step. Those culturing steps performed before DNA extraction and sequencing will actually take more times. So for example, it will take like 1 to 2 weeks time. So therefore, it makes the entire workflow a lot slower and making it for outbreak purpose, the detection and surveillance, it will be quite slow. So therefore, we actually use, let's say, targeted approach like amplicon sequencing to shorten the time required and remove, let's say, the culturing step or making it shorter. Okay. Then also we can use it for food authenticity purpose. Next up, we can use nanopore technology for quasi metagenomics. Basically, here we are again similar to amplicon. We are targeting certain bacteria, foodborne bacteria only. So we already narrow down the targets, and then we are focusing on, let's say, just the target, let's say Salmonella or Listeria. And then we have the entire workflow to focusing on the detection on Salmonella, E. coli, or Listeria only. Again, this particular purpose is to shorten the entire flow from the sample to be questioned until, let's say, the detection of the potential foodborne pathogen. And lastly, metagenomics is basically non-specific. We do not have any targeted, but we're detecting everything inside the sample. So this slide here, I would like to share with you guys that the application of nanopore sequencing technology in the foodborne pathogen detection area. So since 2020, nanopore has been awarded the Food Safety Innovation by the International Association for Food Protection. Okay, so this particular award is because nanopore sequencing has been used for Salmonella and Listeria monitoring. So in actually since 2019, the USDA FSIS already very interested to use nanopore, the sequencing technology for the food safety area. Okay, so therefore they are actively initiate this particular program to use also nanopore technologies for the sequencing of foodborne pathogen. So at the end of 2019, we are able to actually, the scientists in USDA managed to use nanopore technology to perform sequencing and then assemble the genome for Salmonella and E. coli. Okay, so this is the first step ever since long time ago, 2019, to use nanopore in the food safety area. So this is from US and Europe. In 2021, several global daily company already exploring and also investigating the potential of using nanopore technology to monitor spoilage and prevent spore-forming spoilage pathogen in their food production. So especially like Bacillus and Clostridium. Okay, so and also in 2022, FDA researchers are already very interested to use nanopore technology for foodborne pathogen detection purpose, and therefore they actually started. They showing a webcast. They presented a webcast on this particular usage of mean ion sequencing of foodborne pathogen. So nanopore data, sequencing data able to deliver actionable insight in food industry for the purpose of QC monitoring, risk assessment, and recall investigation. So this is showing some scientific community that actually using nanopore and then and exploring the use of nanopore in food microbiology. So in addition to foodborne pathogen investigation, nanopore technology is suitable for the use of food authenticity as well. So basically, we are checking whether there are some contamination of pork residue in the food production, or let's say the whatever food that we are claiming it to be. Let's say we say that it's a very high grade Australian beef, but whether we actually mix with some low standard beef, or even not beef but some other meat to pretend to be the high class meat and selling it for the high price. So this is actually for the food authenticity purpose. Okay. So after looking through some of the companies and also research group and even the research community using nanopore technology on food, and as well as foodborne pathogen detection, as well as the food authenticity. So here I would like to look at some of the more in-depth workflow on using nanopore for those purposes. Okay, so basically we can separate the workflow or approach into two. The first one is whether we actually culture the bacteria from the sample. Okay. The second one is we do not do any culturing, but we do some direct testing. So yeah, this will actually help to reduce the time for detection. So under the direct testing, we have the quasi-metagenomics, and then we have the targeted approach like amplicons using marker like 16S for bacteria, ITS for fungus, and 18S. Okay, so this is showing some of the internal development benchmark against FDA and other mock community. So we have here all the end-to-end workflow for the sequencing of the, let's say, the whole genome sequencing for the isolate, bacterial isolate, direct colony sequencing. So therefore, there will be no additional broth culturing and DNA extraction. Long-read 16S sequencing for the amplicon sequencing, and shotgun is for the non-selective, non-specific sequencing of all the community in the food. Here showing some of the partners of Nanopore that support the use of Nanopore sequencing technologies for food testing purpose. So next up, we will actually go into a slightly more in-depth workflow of using nanopore technology, how we actually use it for the sequencing of the bacterial isolate, or using it for the quasi-metagenomic purpose or non-selective, non-specific sequence or metagenomic methods. Okay, so the first one will be culturing the isolate using whole genome sequencing workflow. So in Nanopore, we have an end-to-end solution that we call Nanopore only microbial isolate sequencing solution. What this particular method produces is basically the whole genome of the bacteria. So in the food industry, it basically means the foodborne pathogen. So what it can offer, the information will be the complete high-quality chromosome DNA information. We can resolve the plasmid, basically means that we can have the entire chromosome as well as a separated entire plasmid. So we can locate the AMR, the antimicrobial resistant genes, whether it's actually reside in the chromosome or it's actually as a mobile element in the plasmid. Okay, so and then we are offering flexible sample batching as well as rapid turnaround time because the library preparation step will be shortened. So here is a study showcasing the reliability of the sequencing technology of Oxford Nanopore. So Dr. Ryan from Doherty Institute basically using a reference strain of bacteria, and then after that extracted the DNA. After that, we sequence using nanopore sequencing technology, assemble the genome. We compare back this data to the reference genome of those reference strain. So we actually have very high accuracy score when during the comparison of this sequencing data. So for, let's say, Campylobacter, it's having nearly a Q50 in terms of accuracy. So therefore, the data generated by Oxford Nanopore is very accurate and reliable. So next up, just now is those some commonly used strain, but here internally in Nanopore, we actually perform sequencing using Oxford Nanopore and then compare to the FDA panel bacterial strain. Again, we actually trying to see the data that produced by Oxford Nanopore, whether it's comparable to what we expected from those reference strains. Okay, so again, we have a Q55 for our sequencing data, and then we have a very high 99.9% mean of CGMS MIST concordance to the reference strain. So we're able to detect the antimicrobial resistant gene and the detected gene sequence are 100% concordance. And for Salmonella, we actually require the information like the serotype of the Salmonella. So based on our sequencing information, we get 100% match with the reference data. Okay. So this is actually an overview of the no-miss solution for microbes whole genome sequencing data production. Okay, so the first day will be the isolate culture into the broth, and then we perform DNA extraction. So it requires nearly a whole day. And then next up, for the preparation of the DNA to become a sequenceable library, we will need just less than 60 minutes. Basically, we have the DNA, we're adding the adapter, and then we do some purification, and now it's actually ready to be sequenced. So for the sequencing, that will take up to 3 days, but a lot of times 1 or 2 days will be enough to generate enough data, more than 50x coverage for the bacteria. So Nanopore is offering the analysis software as well as a free offering. So does not have any additional charge. So it will help to assemble the data generated from the sequencer. Okay, so here is showing the end-to-end solution, the kit preparing the DNA to be sequenceable, as well as the flow cell to perform the sequencing. So next up, we'll look at the workflow for direct from colony microbial sequencing. So compared to just now, we might need to grow the single colony into broth and then do DNA extraction. For this particular workflow, we actually speed up the detection and also identification of Salmonella as well as its serotypes straight away from the colony, okay, without additional culturing in broth as well as the DNA extraction step. Okay, so the information we're able to obtain from this sequencing using the direct from colony sequencing, we'll be able to know the identity, genus species of the Salmonella, the serotyping, the multi-locus sequence typing of the Salmonella, as well as the antimicrobial resistant profiling. Okay. So this is comparable. Salmonella can be processed and then sequenced using the whole genome sequencing method as well. So therefore, we are comparing using, let's say, the standard whole genome sequencing method, which is actually the no-miss. So it will take around 5 days. Whereas if we actually performing this workflow for the direct from colony Salmonella sequencing, the entire workflow only require 1 day. Okay, so you replace method requiring subculture and DNA extraction. So, and then minimum resolution needed for the strain similarity and CGMIST analysis. So here is showing the data for producing from the direct from colony sequencing. So it gives reliable genus and serotyping information. So includes slowly related serotypes, and we are able to differentiate as well as those difficult to serotype organisms from cornea. Okay, so this is showing the reagent required to perform the Salmonella serotyping direct from colony consumables. Okay. Okay, so in a short summary for the whole genome sequencing, we are able to offering the sequencing of the complete genome to give us the gene information like antimicrobial resistant gene, okay, so serotyping, and then it's useful during an outbreak of food QC purpose. And we are offering the direct from colony for the Salmonella only currently. So next up, we have the amplicon sequencing workflow using nanopore technology for the detection of those foodborne pathogens. So here I'm using the 16S marker gene as the example. So the 16S marker gene is actually the marker gene for bacteria. So all bacteria will have this 16S ribosomal RNA gene. The length of this gene is approximately 1,500. So conventional sequencing technology will only sequence a short region of it, which is approximately 400 plus. Okay, so in Nanopore, we actually sequence the entire 16S gene that give us more information and align to the database. We will get a clearer image on the genus or investigating the species of the investigating sample. Okay, so this is showing publications case study that using Oxford Nanopore technology to assess quality and safety of probiotic using the amplicons method. Okay, so the author successfully identified expected the microbes at the anticipated abundance from policy control. Okay. So the study highlighted that 16S sequencing can detect bacterial microbiota even with low abundance in sample. These are some of the additional use of 16S or other targeted microbial identification. Okay, so this is actually by a few research group. So they perform a time point study across manufacturing process, and then they actually map to the specific problematic spots and point specific community. Okay, so this one using Nanopore 16S sequencing technology to check on the supply chain to reveal shift in microbiome between the source and the forwarded produced lettuce. Okay, so again, another time point study of the processed food. So this one is for the storage of cooked rice. Okay, and then this one is checking for the stream disease progression using 16S. So why implement nanopore sequencing for targeted food pathogen detection? So these are some of the advantage of it. Basically, we can actually accurately decide the identity, and we are able to separate the Salmonella serotype very well, and we do not need to perform some removal and isolation step like the bacterial culture and all that. And the entire workflow is very short, and we produce real-time sequenceable data. So this is showing the product from Oxford Nanopore for the 16S sequencing application. So this is showing that we have the analysis workflow available for the 16S sequencing to give you the information of the taxonomy, the diversity, as well as the abundance. So the second last sharing section of today will be the shotgun and the quasi metagenomics. So the advantage of nanopore sequencing for metagenomics will be that we are producing a longer fragment. So after the assembly, we are able to join better, stitch the information better, and give you more information for the analysis of the information. So nanopore sequencing technology enable rapid turnaround time for metagenomics. So the sequencing of food and food environmental sample can be challenging because they actually contains low abundance of the abundant pathogen, causing low abundance of DNA. So at the same time, it has high background from the host or commensal. So therefore, the methods developed for enrichment of microbial DNA. Okay. So this study from Bai Tai is actually has a host removal using the host zero method. And then from Wagner, we actually have a short primary enrichment using the MTA. And then we have other applications showing how to possibly reduce the turnaround time. When we have the metagenomic sample, and then by reducing the pre-enrichment step or shorten those steps, and removal and reducing the sequencing of the host, but focusing on the organism of interest. as only. So this is actually showing a study for microbiome-based environmental monitoring of production facilities. Okay. So the author assessed the performance of a random amplification to overcome low abundance of the microbial DNA load, okay, coming from the environmental sample. So the study highlighted that this approach actually successfully identified some of the most abundant species, including Cocuria, okay, Enterococcus, Enterococcus fausea. Okay, and then this is actually a study to artificially spike in the irrigation water to mimic real-world sample, and then we try to detect the community and see whether we're able to get the abundance as well as the identity of those microbes in the sample. So the shift is microbiome with increasing concentration of E. coli to predict the 0157. Okay. So this is showing the workflow for the metagenomic analysis. So starting from the preparation of DNA, after getting the sequencing data loaded into the epitomy analysis software, and finally the report. So for the last section of today will be the usage of Oxford Nanopore technology for food authenticity purposes. Okay, so we have the sequencing approach and depends on the nature of your sample. So first off, depending whether the food is actually a highly processed food, so we can use the DNA barcoding or metabarcoding using universal primer to allow high discriminatory power. Okay, so for unprocessed food, DNA barcoding or the metabarcoding method is optional. Okay, but not necessary when inhibitors and DNA fragmentation is less prevalent. Okay, so shotgun genomic sequencing is sufficient and easiest and fastest method for unprocessed food. Okay. So here is showing the reason why he implement the sequencing for the metabarcoding in food authenticity. So the method actually overcomes misidentification due to the inhibitors of present in the sample as well as the condition of the DNA, which is highly degraded. So the accuracy and practicality of nanopore sequencing makes metabolic coding promising for fast analysis of complex food and feed mixture. So this is showing the response sequencing workflow for food authenticity purpose and implementing shotgun sequencing for food authenticity. Yeah. Okay, so it's work best with unprocessed food for easiest sample preparation and fast turnaround time. So we can start with, let's say in this example, 50 milligram of pork, then in the mixture, and then we see whether what is the detection. Okay, so with that, I will stop and end my sharing today. Thank you for your attention. So we shall move on to the Q&A session.
Yep. Thank you, Chi Hong, for the insightful presentation about the food testing applications and food safety using Oxford Nanopore technologies, as well as the end-to-end solutions for food industry. Now, we move into the Q&A session. You can unmute yourself to ask question, or you can type your question in the chat box. Yeah. In the chat box, we have a question. What kit would you suggest for WGS, a specific bacteria species, example, Klebsiella, in environmental water samples without the bacteria enrichment or culture method? Do I need to perform any PCR or use barcoding kit too?
Okay, so if you just want to know the identity of the presence of the bacteria in environmental sample, you can actually use the 16S barcoding method. Okay, so you can use PCR amplicons of your own, then use our native barcoding kit. Okay, so we have a kit called native coding ligation kit. So it can work with any PCR amplicon. Okay, or we have another kit that come with the 16S primers and then continue with the shorter workflow compared to the native barcoding kit that I mentioned earlier. Okay. So this toolkit will be enough to give you the information about the identity or the presence or even the abundance of bacteria in your environmental sample. Okay. So this tool set does not need any culturing enrichment because we use a PCR to actually amplify the bacteria using universal barcoding primers, the 16S, 27F, and 1492. And then therefore we are skipping the culturing step. Okay, that will be the good thing, but the downside will be you only get the information about the identity, but nothing else.
Okay. So, is there any question from the floor?
Thank you.
Okay, we have another question. For the metagenomics analysis for fermented foods, are there any specific kits or protocols that you recommend? Would it be the same as for environmental samples?
Okay, so again, depends on the purpose, meaning that what do you expect to get from the sequencing result? If you just want to know the identity, okay, so you can perform a targeted approach, let's say using a universal primer for amplifying your, let's say bacteria of interest or even yeast of interest or some organism that you're interested. Okay, so for the sequencing, basically we do not have like a very special kit for this particular application, but the challenge part will be maybe perhaps the extraction step. So we will be able to recover the microbes or DNA after extraction, and then yeah, we then actually able, if we are able to get the DNA, then we are able to sequence it. Okay, but if let's say for those this kind of like fermented food, there will possibly be remains of the food DNA, I mean the plant DNA there. So therefore, if you're not doing any culturing step or enrichment step, so PCR will always be the best approach to uh, get more DNA, to amplify the DNA from the microbes of interest. Hi, yeah.
Yeah, we have another question from Betty. She is a geologist, and her question is this can be used for metagenomic from fecal samples from animals?
Yeah, so because actually we can use it for this purpose, just that because today my scope is more on about food and food related. So, but our sequencing technology can be applicable to basically any type of metagenomic samples, environmental sample. So, for example, we can actually have gut microbiome, or let's say in your case, fecal sample, as well as soil sample, even water sample. Okay. So we can study all the microbiome metagenomic sample. Okay, let's say across the range that I mentioned above just now. Okay, so microbes. And some that I consider more interesting study will be meta sample from water sample, but they are not looking at the microbes, but they are looking at the foods. I'm sorry, not food, but fish species in the water, the river water. So then they're just using different marker genes to actually focusing on those organisms that they are interested, and then they amplify it and then sequence. We compare to the database to see the identity of what we are interested, as well as potentially the abundance of this organism.
Thank you, Chi Hong. Is there any question from the floor? It, think it seems like all good for now.
I think I can share another interesting research that I came across as well. So just now we mentioned about the guts. So we have another interesting study from one of our researchers. Basically, they study the gut of the fish, but they are not looking at the microbes, but what kind of fish, the food, what these big fish are eating those small fish. So we are amplifying the gut content and then to see what kind of fish being eaten by this large fish as well. Yeah.
Wow, that was interesting. Yeah, I think there is no more question from the floor, so we will end the webinar for today. Again, thank you, Chi Hong, for the informative and engaging presentation for today. And a big thank you for you all to those who are participating in this webinar. And we hope you find the information shared here is valuable and can inspire you to apply it to your own research. Before we end the webinar today, we would like to hear your thoughts. Please take a moment to fill out the survey form in the chat box. And if you have any question after this, feel free to contact us at sales@nextgene.my, and our representative will reach out to you. Thank you all for once again, and we would like to look forward to connecting with you in the future events. Bye bye.
Thank you. Bye.